I'm sure you've heard of starfish. They're easy to identify as most have 5 arms looking like a star. Even though we find them in every ocean, the name is misleading as they are not fish at all. Starfish, sometimes called sea stars, belong to the phylum Echinodermata. The name comes from the Greek and means prickle-skinned. Other members of this phylum include the brittle star, sand dollar, sea urchin, and sea cucumbers. The starfish has a hard, radially symmetrical body, covered with spines which radiate into 5 tapering arms. These invertebrates are restricted to the marine environment, yet have been collected both from the shallowest pools and from great ocean depths. While having a lifespan of 3 to 5 years, they possess the unique ability to regenerate body parts. In fact, if a starfish was cut in 2, each half would form a new starfish. Watch now as we demonstrate the recommended procedure for dissecting the starfish.
To start our study of the external anatomy of the starfish, we've placed our specimen dorsal side up in the dissecting tray. Note that we have 2 specimens that are pretty much the same, with the exception that this one has been dyed so that we can see some of the internal organs better. To start our external anatomy study, we'll note that there are 5 rays or arms on the starfish. If we look on the central area of the starfish, slightly off center, is the sieve plate, which is also referred to as madreporite. This will draw water into the starfish. If we stroke the skin of the starfish, we note that it has a spiny texture, and that is due to the fact that it has the presence of spines, and it is in the phylum Echinodermata. If we look on the ventral side of the starfish, in the central area there's this opening which is the mouth. If we go from the tip of the ray all the way to the center, we see that there's a groove, and on either side of that groove we see 2 rows of tube feet. These tube feet will be used to cling onto a clam as it tries to pry that clam open to feed upon it. To begin our dissection of the starfish, we need to snip off an end of the ray and cut through the skin along each side of the ray. Then once we've cut up along each side of the ray, we can lift this up. We want to cut around that circular disc area and just cut through the skin so as not to damage any of the internal organs. Now we can identify the green digestive glands, the stomach, and if we remove the digestive glands, underneath these organs we can see these granular jelly-like structures which are the gonads. Now we're ready to study the framework of the starfish. Going down the ray of the starfish is this ambulacral groove, and extending from the ambulacral groove are these skeletal ridges. Also along the ambulacral groove are these swollen areas, bulb-like structures referred to as ampullae. To study the water vascular system, we'll need to remove the stomach, the water vascular system consists of the madreporite that we've already pointed out. Water comes in through the madreporite, will go down through a very small canal, and I have a specimen that shows this a little bit better. On this specimen we can see here's the madreporite again, and then here's a stone canal that goes down from the madreporite to this circular canal, which is known as a ring canal. Water goes through the ring canal and then down through the radial canal. Since the starfish has 5 rays, there would be a radial canal in each ray. From the radial canal are these bulb-like structures that we've already pointed out, which are called ampullae. The ampullae hold the water until the water goes down into the tube feet, which are on the ventral surface again. On our specimen that has been injected, we can see that the tube feet are quite extended here. To study the digestive system now, I'm going to refer back to our other specimen. And in the central area now, we see the stomach. And when the starfish feeds, what it does is it uses its water vascular system to grip onto a clam and to pry it open with the section of its tube feet. And then the stomach is going to be ejected out through the mouth. On this other specimen, we can see that the stomach is extending out of the mouth. This stomach then will go inside the clam shell, digest it, and absorb the nutrients. The digestive gland is this green structure that makes the enzymes for the digestive juices. Since the digestion takes place externally, there's a very poorly developed intestine, which would be on the dorsal side, and also poorly developed rectum and anus. The reproductive system consists of gonads, which are found in each ray underneath the digestive glands. In this specimen, we can see that there are these granular, jelly-like structures making up the gonads. Now, we refer to these as gonads because we're not sure whether this is a male or female. To identify the sex of it, we'd need to take some of the gonads out, put it on a microscopic slide, and identify whether they're sperm or eggs. In the dyed specimen, I have one that has already been dissected. We can see that the gonads show up red in this specimen. I've not pointed out a heart or blood vessels because a starfish does not have a circulatory system, meaning that it does not have a heart or blood vessels. Rather, circulation just takes place by water flowing through the body cavity, carrying nutrients, oxygen, and also removing carbon dioxide. This concludes our dissection of the starfish.
The dissection of the starfish illustrates the unique characteristics of the great marine phylum Echinodermata. Spend time with your specimen, as it is a descendant of a starfish that crawled over the ocean floor millions of years ago.